But ancient DNA has shattered that myth. Today, scientists can read the genomes of humans who lived tens of thousands of years ago, and what they've uncovered tells a story far more complex than anyone ever imagined.
Everything you have been told about race is a lie, and the DNA in your own cells is the evidence that proves it. For centuries, we have categorized humanity into neat, distinct boxes based on geography and skin tone, assuming these labels reflect fundamental biological divisions. But when researchers finally mapped the full spectrum of human genetic history, they found something that shatters our entire worldview. One specific group reveals a biological reality that defies everything we thought we knew about human ancestry. The facade is crumbling, and the data suggests we have been looking at our own history entirely backward. The myth of biological race. Everything you have been told about race is a lie, and the DNA in your own cells is the evidence that proves it. We have spent centuries categorizing humanity into neat, distinct boxes based on geography, skin tone, and facial features. We were told these labels reflect fundamental, deep-seated biological divisions, that there is something inherently different about the genetic blueprint of a person from one part of the world compared to another. But when we look at the human genome at the level of base pairs, that entire facade crumbles. It turns out the biological basis for race is a mirage. Think about our species in the context of the natural world. If you compare two chimpanzees from different forests, you will often find more genetic variation between them than you would find between two humans chosen from opposite sides of the globe. Humans are among the most genetically homogeneous species on the planet. Our genome consists of approximately 3.2 billion base pairs, and across every single person on Earth, we share 99.9% of that code. That tiny 0.1% difference is responsible for the vast array of individual traits we see, but it is not arranged in the discrete, tidy packages that people have tried to call races. We are, at our core, a species defined by recent common ancestry and a relentless history of migration. The most damning evidence against the concept of biological race emerged in 1972, when a Harvard evolutionary biologist named Richard Lewontin changed the way we understand human diversity forever. Lewontin performed a meticulous analysis of blood types, protein variants, and genetic markers across various populations. He wanted to know where the variation actually lived. What he discovered was a mathematical wrecking ball to the idea of racial taxonomy. He found that roughly 85.4% of all human genetic variation exists between individuals within the same local population. Only about 6.3% of the total variation could be attributed to differences between the conventional races we recognize today. Modern genomic studies, utilizing massive datasets and the most advanced statistical tools available, have only confirmed his findings. Some of the most recent estimates suggest the variation within a local group might be as high as 86%. This means that two people from the same so-called racial group can easily be more genetically distant from one another than either of them is to a person from a completely different part of the world. Mathematically, racial categorization fails to capture the reality of our biology. It is a system that tries to define the species by the smallest sliver of our total genetic makeup, while ignoring the massive overlap that defines our shared humanity. To make matters worse, the physical traits we use to define race, like skin pigmentation, are what scientists call discordant. They are inherited completely independently of other vital genetic markers. Skin color is a polygenic trait, heavily influenced by historical environmental exposure to ultraviolet light. However, if you map out skin tone, it does not align with the map of your ABO blood types, your susceptibility to certain diseases, or your ability to digest lactose. If we decided to classify humans based on whether they could process dairy or the specific pattern of their fingerprints, we would create entirely different groups of people. Choosing skin color as the primary marker for race is a completely arbitrary decision. It is a superficial categorization that holds no more biological weight than grouping people by their favorite color. The scientific consensus today is firm. Race is a social and political construct, not a biological reality. It is a label that has been used historically to build hierarchies and justify inequality, but it does not describe the continuous, clinal nature of human variation. Humans do not exist as distinct branches on a tree. We exist as a smooth gradient of genetic flow across a global landscape. There is no deep, branching structure in our DNA that supports the existence of biological subspecies. When we lean on these outdated racial labels in medicine or social policy, we are often working with a flawed map that causes real-world harm. We are a species defined by constant mixing and movement, and the attempt to force that complexity into rigid boxes is a relic of a pre-genomic age that the evidence has long since left behind. Evidential issues of ancient genetic mixing. For generations, the story of human origins was painted as a clean, predictable line. We were taught the out of Africa narrative. A singular small group of ancestors emerged from the cradle of humanity and marched across the globe, gradually refining into the modern humans we recognize today. It was a neat, linear progression, easy to map and simple to visualize. But that version of our history is becoming increasingly difficult to defend. The latest genomic data tells a far more turbulent and fascinating story, one that looks less like a single, solitary path and more like a braided river of ancient, divergent populations that spent hundreds of thousands of years separated before finally colliding. To uncover this hidden past, scientists at the University of Cambridge stopped trying to rely solely on the fragile, often decayed DNA found in fossilized remains. Instead, they built a digital time machine. They developed the COBRA algorithm, a sophisticated coalescence-based hidden Markov model. This tool acts as a high-powered lens that peers into the architecture of modern human DNA to reconstruct ancestral splits and reunions. It can identify the footprints of ghost populations, groups of ancient humans who left no physical remains, yet whose genetic influence remains woven into our cells to this day. The results generated by this model are nothing short of a paradigm shift. COBRA reveals that the blueprint for modern humanity was formed by the merger of two distinct ancestral populations that had been isolated from one another for an astonishing 1.5 million years. Think about that time frame. For over 1 million years, these two lineages followed entirely different evolutionary paths, developing in separate corners of the world and facing different environmental pressures. They were so distinct that, in any other context, we might have classified them as separate entities. Then, roughly 300,000 years ago, these two branches collided in a monumental admixture event. This merging was not a perfectly equal exchange. The data reveals a distinct 80-20 split. 80% of our modern genetic heritage can be traced back to the primary majority lineage, while the remaining 20% acts as a silent, mysterious companion. What is truly compelling is where this 20% hides. We find this genetic legacy primarily in the non-coding regions of the genome, the vast, mysterious stretches of DNA that do not carry the instructions for proteins, but instead orchestrate the complex regulatory functions of our biology. This specific distribution suggests a process of purifying selection. Because that 20% of foreign, ancient DNA was so different, natural selection had to act as a filter, systematically scrubbing away any mutations that were incompatible with the 80% majority, while retaining the segments that offered unique regulatory advantages. It is as if our cells underwent a massive, million-year-long integration project to ensure these two divergent, ancient histories could actually function together. The majority population itself appears to be the same lineage that eventually gave rise to the Neanderthals and the Denisovans, meaning our history is not just connected to these other ancient groups, but deeply, inextricably tangled with them. This discovery moves us away from the idea of humans as a stagnant, linear species. Instead, it reveals a history of deep, ancient interbreeding that is embedded in every living person today. The Cobra algorithm has even been used to apply this same logic to bats, dolphins, and our closest primate relatives, proving that this complex, braided history is a common feature of life on Earth. We are not the result of one single, pure line of descent. We are the survivors of a grand, biological convergence, a product of ancient populations that crossed paths after eons of isolation to create the unique, complex, and deeply interconnected species we are today. Our DNA is not a static record of a single ancestor, but a living archive of a massive, multi-million-year collision of worlds, the legacy of genetic drift and bottlenecks. In 1775, a devastating force of nature known as Typhoon Lenkieki swept across the Pacific, obliterating the Pingalap Atoll in Micronesia. The storm was cataclysmic, leaving the island a ruin and reducing the population to a mere handful of survivors. Only 20 people remained to rebuild their world from the ashes of disaster. Among that small group was the island ruler Doakesa Mwananiset, who carried a secret in his genetic code, a rare recessive mutation for achromatopsia, or complete colorblindness. In the isolation of the following generations, that one tiny, random act of survival became a biological blueprint for the entire population. Today, 10% of the people on Pingalap are born with total colorblindness, a condition that usually appears in less than 1 out of every 30,000 people globally. This is the chilling, tangible power of a genetic bottleneck. When a population is suddenly decimated, the survivors take with them only a fraction of the original gene pool. Whatever traits happen to be present in those few survivors, whether beneficial or completely neutral, become the new foundation for the future. The case of Pingalap highlights that evolution is not always about the survival of the fittest. Sometimes it is simply about the survival of the luckiest. Genetic drift is the engine behind this phenomenon. It describes how the frequency of gene variants can rise or fall over time purely due to random chance, rather than any specific survival advantage. In large populations, these random fluctuations are diluted, but in a group of 20, they are explosive. A single carrier can change the destiny of an entire community within just a few generations. On Pingalap, this drift resulted in a condition known as rod monochromatism, where individuals lack working cones in their retinas. They live in a world without color, experiencing extreme light sensitivity and involuntary eye movements. In a striking clinical anomaly, 85% of these individuals even exhibit a paradoxical pupillary response. When they move from light into darkness, their pupils constrict instead of dilating. This isn't an adaptation to the environment, it is a permanent mark left by a single, catastrophic typhoon centuries ago. We often look at regional genetic differences across the globe and assume they must be adaptive, that people in different regions evolved specific traits because those traits provided a survival advantage. We look at skin tone, blood types, or metabolic differences and search for a reason why they are better suited for that local environment. But the reality is far more grounded in happenstance. While natural selection does drive some profound changes, a vast majority of the regional genetic variation we see in humans is essentially neutral. It is the result of founder effects, bottlenecks, and the simple, messy accumulation of mutations over thousands of years of migration. Our DNA is more of a historical diary than a survival manual. Each marker in our genome acts as a trace of where our ancestors traveled and when they encountered one another. When small groups moved into new territories, they brought their own random assortment of genes with them, which then spread throughout their new, isolated populations. These variations do not necessarily confer a biological edge. They are simply the signatures of our shared and divergent journeys across the planet. When you strip away the narrative of adaptation, you are left with a clear picture. Humans are shaped by the accidents of history as much as by the pressures of nature. The Pingalapese experience serves as a stark reminder that human diversity is often born from isolation and randomness. We are the products of millions of small, unplanned events, and most of the regional differences we use to tell our stories are just markers of the paths our ancestors happened to take. We are walking records of a long, chaotic, and fascinating history, defined by the luck of those who came before us. Controversial genetic adaptations. Evolution is usually a slow, indifferent process, but there are moments when the environment turns up the pressure so high that life is forced to pivot. These are the moments where biology stops being random and starts being surgical. We are not talking about superficial traits or the results of ancient accidents. We are talking about genuine survival superpowers. When a population faces a life or death challenge, whether it is a lack of oxygen, a new source of food, or a deadly parasite, the human genome has the remarkable ability to innovate. Look at the way we digest milk. Most mammals, including the vast majority of our own species, are lactose intolerant as adults. We lose the ability to break down milk sugar the moment we are weaned. But in cultures with a deep history of cattle domestication, something incredible happened. Both in Northern Europe and in parts of East Africa, human populations independently evolved the ability to keep the lactase enzyme active into adulthood. This is a masterclass in convergent evolution. Europeans achieved this through the 13,910 *T allele, while populations like the Tutsi and Fulani in Africa evolved entirely different genetic solutions on their own unique backgrounds. This was a massive win for survival. Those who could digest dairy had a reliable, consistent source of hydration and nutrition, providing such an advantage that it resulted in up to 19% more children per generation. If that sounds impressive, consider the Tibetans living on the high-altitude Tibetan Plateau. Most people who travel to such extreme heights develop thick, viscous blood and cardiovascular issues because their bodies are struggling to adapt to the thin air. But the Tibetan people thrive. They carry a specific version of the EPAS1 gene that acts like a biological high-altitude filter. The most astonishing part of this story is that this gene was not a brand new invention. It was a genetic gift acquired through introgression from the Denisovans, an extinct human relative about 43,000 years ago. This ancient, archaic code sat dormant in the population for millennia, serving as a silent backup until the permanent settlement of the plateau 12,000 years ago, when natural selection surged and drove this trait to an 87% frequency. It is a stunning example of how ancient interbreeding provided the raw material that allowed modern humans to conquer the highest places on Earth. Our diet tells a similar deep time story. Consider the AMY1 gene, which produces salivary amylase, the enzyme we use to break down starch. Chimpanzees generally have only 2 copies of this gene, but modern humans can carry up to 17. While we often associate this with the invention of agriculture 10,000 years ago, the process actually began much earlier. Genetic evidence suggests these duplications were appearing as far back as 800,000 years ago, showing that even our early hominin ancestors were learning to harness wild starches as a fuel source long before they ever picked up a plow. These adaptations, From malaria resistance in the Mediterranean and West Africa to the high-altitude biology of the Himalayas, are localized survival responses. They are functional, cold-blooded engineering projects designed to keep a population alive in a specific niche. They have absolutely nothing to do with the social categories we call race. In fact, these traits are often found in completely different parts of the world, appearing wherever the pressure of the environment demanded a specific answer. When you view our history through the lens of these adaptations, you see a species that is remarkably fluid, constantly borrowing and building the tools it needs to survive. We are not a collection of distinct races defined by static biology. We are a species of master adapters, using every tool in our genetic library to navigate a challenging world. Ethical and social controversies. We are living in an era where we can read the blueprint of life with terrifying precision, but this power comes with a dark, persistent shadow. History shows us that whenever humanity thinks it has decoded the secret to our value, we find a way to weaponize that knowledge. Look back at the early 20th century, a time when figures like Sir Francis Galton and Charles Davenport turned the study of heredity into a tool for state-sanctioned cruelty. They called it eugenics, a movement obsessed with the impossible goal of improving humanity through selective breeding and forced sterilization. Laws were written in the United States to systematically strip the rights of those deemed unfit, a model that was soon adopted and scaled to industrial, genocidal proportions during the Holocaust. That dark chapter should serve as a permanent, flashing warning light for anyone who thinks they can use genetics to justify a social hierarchy. That danger has not vanished. It has simply changed form. Today, it hides in plain sight within our medical institutions. Consider the EGFR formula, the standard calculation doctors used for decades to estimate kidney function. For years, this formula included a race-based variable that automatically boosted the score for Black patients, under the flawed assumption that they naturally had higher levels of creatinine. This was not just a minor inaccuracy, it was a systemic failure that actively harmed thousands of people. By overestimating the kidney health of Black patients, the medical system systematically delayed their access to life-saving transplant lists, medication, and specialized care. It was not until 2021 that the American Society of Nephrology and the National Kidney Foundation finally discarded these race-based equations. This simple change opened the door for over 31,000 more patients to become eligible for transplants, proving that racialized medicine was not only inaccurate but lethal. This catastrophe is a symptom of a much larger, more pervasive threat: the destiny fallacy. This is the widespread dangerous belief that our lives, our intelligence, our criminality, and our socioeconomic outcomes are written in stone by our DNA. When we fall for this genetic essentialism, we treat social problems like poverty or educational achievement as if they were biological inevitabilities. In reality, human traits are incredibly complex and polygenic, deeply influenced by the environments in which we develop. Studies on intelligence, for example, consistently show that socioeconomic status plays a massive, often dominant role in determining success. When we treat genes as a totalizing script for a person's life, we are essentially building a new, sophisticated justification for inequality. The threat is compounded by the commercialization of our very identity. Millions of people are handing over their genetic data to private corporations that treat our ancestral history as proprietary property. Once that information is in the hands of a company, the question of how it is used, who sees it, and how it might be interpreted remains largely in the dark. We are essentially giving away the deepest, most private code of our existence to entities that prioritize profit over the ethical responsibilities of scientific stewardship. Researchers now face a critical moment of accountability. Organizations like the National Academies of Sciences, Engineering and medicine are sounding the alarm, demanding that we stop using race as a shortcut for genetic variation. It is an intellectual and moral failure to rely on these social labels when the actual science shows us a continuous, interconnected, and clinal reality. Science is a tool, not a weapon of validation for our biases. If we are to honor the truth revealed by our own genomes, we must defend the dignity of every individual against the seductive, dangerous lie that our blood determines our worth. The responsibility to keep this science grounded in humanity, rather than in the service of old prejudices, belongs to all of us. The 1000 Genomes Project and its issues. For most of human history, our understanding of ourselves was dangerously limited by what we could see on the surface. We looked at a person and made assumptions based on geography. We categorized based on appearance and skin tone, eventually building entire social hierarchies and medical frameworks on the shifting, unstable sands of outward phenotype. We mistook the cover of the book for the entire story. But in 2008, the global scientific community launched an effort that would finally shatter those ancient, inherited myths. They began the 1000 Genomes Project, the most ambitious, costly, and comprehensive map of humanity ever attempted. It was a massive, collaborative undertaking that sought to move beyond the superficial and decode the full, unvarnished spectrum of our biological reality. By the time the project reached its final phase in 2015, scientists had sequenced the genomes of 2,504 individuals representing 26 distinct populations from every corner of the planet, effectively creating a high-definition mirror for our species. The numbers alone are staggering and speak to the sheer scale of the endeavor. This team discovered and cataloged more than 88 million genetic variants, a monumental digital library that includes over 84 million single nucleotide polymorphisms. It was the first time in history that scientists were able to detect genetic variants that appear in as little as 1% of a population, providing a level of resolution that had previously been considered unthinkable. This data became the new gold standard for medical and evolutionary research. It provided the bedrock upon which modern genome-wide association studies now stand, allowing us to pinpoint the genetic basis for common diseases and trace the complex, winding, and often surprising roots of our collective human migration across the globe. To handle this massive, unprecedented influx of information, researchers had to invent an entirely new language of discovery from scratch. They developed sophisticated bioinformatics tools like VCF Tools, Tabix, and the Ensemble Data Slicer, which allowed them to navigate the billions of base pairs that define the human experience. These tools did more than just count genes. They allowed for genotype imputation, a revolutionary process that effectively predicted an individual's genetic makeup even for regions not directly captured by sequencing. This was the precise moment science moved from theoretical guessing and ancestral folklore to absolute, data-backed mapping. The 1000 Genomes Project did not just show us our variations, it showed us exactly how those differences are distributed, confirming the out-of-Africa expansion and scientifically proving that the greatest concentration of genetic diversity resides within the populations of Africa. When you look at this data, the old concept of discrete biological races simply vanishes. You do not find the hard, jagged lines that separate the human species into different subspecies or distinct biological silos. Instead, you find a beautiful, unbroken, and fluid gradient of variation. You find a story of constant, restless movement, ancient intermingling, and a degree of homogeneity that is deeply humbling. We share 99.9% of our DNA with every other human on Earth. This is not just a polite statistic or a convenient talking point. It is the fundamental, inescapable biological truth of our existence. We are a single, deeply interconnected species, shaped by a history of resilience and adaptation that far outweighs the minor, superficial variations we have spent so much time obsessing over. As we look toward the future, the data from this project continues to be our greatest tool for progress. It is helping us move toward a new age of medicine that treats patients based on their individual biology rather than outdated, arbitrary racial labels. It is providing a foundation for us to understand our health, our history, and our future with an accuracy that was once the stuff of science fiction. The 1000 Genomes Project has provided the final, definitive answer to the long-debated question of human classification. The data shows us that our differences are not barriers. They are simply the markers of a vast, shared journey. Our greatest strength is not in our perceived divisions, but in the overwhelming, biological reality of our unity. The record is clear. We are one people, one history, and one future, linked by a code that tells the story of our success as a species. The myth of race is over. The reality of our interconnection has only just begun.